So before we cover chromosomal genetics, let's go back to meiosis and Mendelian genetics and make sure we're really clear on how alleles are passed down to offspring. So let's say we're going to do a cross, and we're going to cross a red flower with a white flower. So this is a monohybrid cross. We're going to follow just one trait, and that trait is flower colour. And there are two alleles. So big R, we're going to say, is red, and so that's dominant. And little r is white, and that's recessive. So to be a white flower, you basically don't have any working versions of the gene that says be red, so the allele that's red. So you have to be homozygous, rather, recessive. So this flower here is going to be homozygous recessive. This flower here could either be big R, or big R, little r, because you just need, in normal Mendelian genetics of normal complete dominance, you need just one version of the working allele to cause a red flower, to cause that allele to be expressed in the phenotype. But really importantly, the reason why I'm writing this down is because I want us to look at the chromosomes. So every organism that reproduces sexually has two chromosomes of every version of the chromosome. So chromosome number 1, it has two of them. Chromosome number 2, it has two of them, etc., etc. So we find the chromosomes in homologous pairs because one came from the male and one came from the female. So this is what the chromosomes look like for this individual. All right, this flower here, if it's big R, it has one chromosome, and at the gene location that says make flower color, it's got a version that causes red flowers. And here on the other version of the chromosome, it has a big R. So this one came from the mother and this one came from the father. If it's this genotype, then it has one of the pair says make red flowers, and the other chromosome, which is homologous, I'm just drawing it like this so you can see the difference, is a recessive allele on it, which basically doesn't work. This organism here has two chromosomes, one like this and one like this. Right. Now it's important to look at these because when we do a Punnett square, you'll notice, let's say we're going to cross this one here with this one here, right? And change the colour. So that would be little r on the top, and then big R here. The reason why we're doing this is because, remember, in meiosis, these homologous pairs of chromosomes are separated. Let's take this organism through meiosis. In fact, let's do this one. This one has two different versions. So this is the pair of chromosomes here. So this organism starts out like this, big R, little r, and this is going to be G1 of interphase because we haven't got those X shapes yet. It then goes through S phase, and the chromosomes we get that X shape. We've got the duplicated chromosomes. So this one is big R here, and then this one is going to be duplicated like this. So we've got the little r, and then the duplicated one there, little r. Okay, so this is the cell after S phase. Now remember what happens in metaphase 1 of meiosis is that these pairs are they're going to line up in pairs like this, and then the pairs are going to be separated like that. So at the end of meiosis 1, we end up with one cell that's got this chromosome in, big R, and we've got one cell that's got this chromosome in here, little r. So notice that only one version of the pair, either the big R or the little r, has ended up in these daughter cells here. Now each one of those then go through meiosis 2, and in meiosis 2 it's just the same as mitosis, you're just separating these sister chromatids. So at the end of meiosis 2 we're going to have one cell like that, one cell like that. Those came from this one here, and then this one's going to split in half, and we're going to have little r, little r. So that is why we draw with the Punnett square. The Punnett square for this one would be big R, little r. Those are the two possible gametes. I know we have four gametes here, but notice how there's only two versions. So we could do this Punnett square where we did a big R, and then little r, but it would be pointless. So that is why we draw it like this, and we have the chromosomes. We only have one version at the top. So this cross here, big R with little r, would produce all offspring that are heterozygous. So be very, very clear on what's the link between the genotype and basically this Punnett square, which is just showing the gametes. What gametes, which versions of alleles can be passed down to individuals when they reproduce? The really, really important thing is that only one version of the gene gets put into the gamete. Every organism that reproduces sexually has two versions of every gene, okay, or two copies of every gene. It could be the same version, like this organism here has two copies of the gene, but it has the same version copied twice, or it could be this individual here that's got two different versions, okay? So, for example, if we have a bird that's got, it's heterozygous for colour, and it's got a gene that causes blue, and it's got a gene that causes green. When it makes gametes, some of the offspring will inherit big B, and some of the offspring will inherit little b, okay, depending on which egg is fertilized or whatever, and then the male will do the same thing and bring in the other version, the other pair. So that's really important to understand. Make sure you're very clear on gametes and how they form. All right. Now let's look at gametes for a dihybrid cross. So let's look at an example of colour where we've got leaf size and flower colour, right, where we've got big L equals large leaves, little l equals small leaves, and then big R is red flowers, and little r is white flowers. So let's say we cross two organisms, because that's what we're doing. We're crossing a male and a female, and we're going to do a dihybrid cross. So we're going to follow both leaf size and flower colour in the plant. And the plants have many, many, many traits, but the important thing is that we're going to ignore everything else. Let's say there's also fruit colour. We're just going to ignore that. We're only interested in following these traits here. So let's just take I'm just going to make up a cross. So I'm going to have big L, little l, and then, so that's one pair of chromosomes, and then big R, little r, that's one organism, and we're going to cross it with one that's little l, big R, little r. Okay, let's draw the chromosomes for these organisms. That's the really, really important thing to do. So I haven't mentioned anything about where these genes are located. Let's say they are located on different chromosomes. That means for this organism, it's going to have, remember, it's got two versions of every gene. It's got one pair of chromosomes that's going to have the gene for leaf size on. We've got a big L and a little l. And then it's going to have another pair of chromosomes, which is a different pair of chromosomes, that's going to have flower colour on like that. All right, so this is what the chromosome is going to look like for this organism, and this organism is going to have little l, and then big R, little r. All right. So what you need to do is you need to figure out the gametes. Now, Mendel's law of independent assortment means that which version of every gamete, which version of the gene one gamete gets for leaf size has no bearing on which version of the gene the gamete gets for flower colour. So if a gamete gets, for example, this chromosome there, so big L, whether it gets big R or little r is completely independent of which L it got. So it could either get big R or little r. So let's say it gets little r. That is one of the possible gametes that could be made from this organism. Remember that the gametes are separated. Right. Just as likely it could make a gamete that's got little l and big R, or little l and little r, or big L and big R. Now let's just be really clear on that and draw out these chromosomes going through meiosis. So you've got one pair, it's got big L, little l, and you've got one pair that's got big R, little r. Through S phase, the chromosomes start looking like this: big L, little l, and then the R is going to look like this: the big R that duplicates, big R, and then you end up with the little r like that. Now, in meiosis 1, remember that the pairs line up next to each other. So the big L and the little l, these homologous pairs are going to line up, and then the pairs are going to be separated like this. Okay? So after meiosis 1, we're going to have a cell that's going to have these two in. So it's going to have big L, and it's going to have big R like that. And then this one here is going to have little l, and little r. So the gametes that are made from these are going to be little l, little r in that case. Right, and this one's going to be big L, big R, because remember these are going to be separated. Just as easily, we could have had it so the R's lined up in a different way. So when they lined up in their pairs, we actually ended up with little r on this side and big R on this side. And we started off the other way around in the other example. So then the gametes would be big L, little r, and little l, big R. So these are the possible gametes that can be made, and it's all because of meiosis 1, metaphase 1 of meiosis 1, how these guys line up, whether big L is on the right or on the left, and then the same here. It's completely random every single time, and that's why we end up with the version of the L that you get is independent of the version of the R that you get. How these line up next to each other, whether it's big L on the left or big L on the right, has no bearing on whether big R is on the left or the right on here. Okay, I know that was a bit long-winded, but hopefully you can see what's going on there. Okay, so let's do this cross. Let's figure out the possible gametes that could be made. And remember, it's all about possibilities. It doesn't mean that this is exactly what's going to happen. It means that if we cross these two organisms, what are the potential offspring that could be made? So here's one organism here. Let's say this is the female, and let's say this is the male. So the female is going to make eggs, and every time she makes eggs, she's going to put one version of the L chromosome in the egg and one version of the R chromosome, along with all the other chromosomes, but remember, we're just following these. So possible gametes that this organism could make, we can do the FOIL method. So I'm not going to do the FOIL method, I'll do it over here in a bit, but I'm just going to follow it more logically. So some gametes are going to have big L and then this version of the R, big R. Some are going to have this big L, but then this version of the R chromosome, the little r. And then some are going to get this version of the L chromosome, so small leaves, and they're going to get the red flower version here. And then others can get the little L, but they could also get white flowers. So these are the particular gametes that could be made by this organism. Okay? And let's do the FOIL. So you do, I have to write it out. FOIL. So first, and then outer, and then inner, and then last. And you see that those are written down there. But we got the same answer. Now this organism, it could put this one, this chromosome, and this chromosome here, so little l, big R. It could put this one here, little l, and then little r. And then it could do this one here, little l and big R, and little l and little r. Now notice how we've got the same, we've got a repeat because we've got the same, it's homozygous here. So when we're doing a Punnett square, we don't actually need to write these out as well. So possible gametes from this one at the top. Now let's look at the gametes for this one. You've got little l, big R, and you've got little l, little r. So if this gamete is fertilized by this gamete here, you're going to have this chromosome and this chromosome of the pair, so big L, little l, and then big R, big R. Here you're going to have big L, big R, little r. Little l, big R, big R, little l, big R, little r. All right. Now remember I said that there were duplicates down here. I could add the same here, but all we'll be doing is copying this line, so it's like you're doubling the probabilities. It works out exactly the same. Okay, so you can do the 4x4 Punnett square, or you could just do it like this. Let's just look at these gametes here. All right, so if we say what is the probability of getting a flower, a plant that's got red flowers and small leaves, it's gonna be, so red flowers and small leaves, so it needs at least one big R, and it needs to be little l, little l. Yeah, so it's out of 8. So big R and then little l, little l. So little l, and a big R, there's 1. Okay, big R, 2, 3. Okay, 3 out of 8. This is why I like doing the branch diagrams, because can you see how it's easy to miss one? If you do the branch diagram, it's super easy. You end up doing it out of 16. Okay, so the branch diagram would end up with 6 out of 16, which is 3 out of 8. It's the same thing. Again, if I added these, duplicated this here, we would have 16 organisms, offspring, possible offspring, and we would have this twice, so we would have 6 out of 16. So you can see what I'm saying, you only need to do this once. You don't need to add double rows if you end up with just 2 versions of the gametes. I hope I'm making sense. If not, just completely ignore me and just do the 16 Punnett square. Okay, you'll get the same answer. Right, now we've figured out how alleles are normally passed on with one version going into every gamete. Let's just talk about linked genes, and this is where things can get confusing. However, if you just kind of think about it logically and follow it through, you'll find that it's not actually that complex. All it means with a linked gene is that they're on the same chromosome. So normally, remember when I showed you the chromosomes of an organism that was, for example, big R, little r, big L, little l, the chromosomes look like this if they were on different chromosomes, if the genes were on different chromosomes. So you've got one pair is going to have the big R, little r, like that, and then the other pair is going to have the leaf size, it's going to have big L, little l. Now, if the genes are linked, they're on the same chromosome. So the gene that causes flower colour is on the same chromosome as the gene that causes leaf size. And so you would end up, instead of 4 chromosomes in 2 pairs, you end up with 1 pair of chromosomes. Okay, so 1 pair. And here you're going to have like the little r and the little l. And in an example, if I in an exam ask you to talk about linked alleles and I give you a heterozygous genotype like this, I'm always going to tell you which versions are on which chromosomes. So is it big R and big L, or is it big R and little l together? And I'll usually give it with this example here. All right. So let's do a cross of individuals two organisms that have got genotypes in which the genes are on linked chromosomes, they're linked genes, they're on the same chromosome. So let's cross big R, big L with little r, little l, little l. Let's do this cross. Now, normally the gametes that we would get from this organism would be, okay, we get maybe this big R with this little this big L, and then this big R could go with this little l, etc., etc. But remember that now the chromosomes, there's only one chromosome, the genes are on the same chromosome. So let's assume that this is what the chromosomes look like for this individual. Okay, the big R is on the same chromosome as the big L, right? Not the little l, but the big L. So when the gametes are made, remember homologous chromosomes are separated. So half the gametes are going to get this one, this chromosome, and half the gametes are going to get this chromosome. So notice how there are only two possible gametes now, two possible combinations. There's either big R, big L, this chromosome here, or there's this chromosome here, little r, little l. All right, now I'm going to talk about crossing over in a bit, so let's assume no crossing over has happened. And if you're not sure what I'm talking about right now, just ignore it, just assume that these are the chromosomes. Now this organism here, the chromosomes are like this, big L, little l, little l, little r, little r. Okay, so it can only make a gamete with either a little r or a little l. That's all this one can make. And when you're given this example in an exam, it's actually super easy because, well, this is the Punnett square that you need to do for this cross. This is going to give little r, little l, and nothing else. Can't give anything else. That's all it's got. So the possible offspring from this cross, considering that the genes are on the same chromosome, they are linked genes, are going to be big R, big L, little l, or little r, little l, little l. Okay, so these are the two possible combinations that you can get. From this cross here, they're going to look 100% like the parents. The parents were a plant with red flowers and large leaves, or with white flowers and small leaves. And the offspring are going to look the same. These possible offspring are going to have red flowers and large leaves, and these are going to have white flowers and small leaves. That is with no crossing over. So assuming linked genes with no crossing over, if we do this cross, all the offspring are going to look like the parents. Notice how we have not got any possible offspring that have, for example, red flowers but small leaves, or white flowers and large leaves. That's really important to note at this point. Okay, so from the cross, big R, big L, little l, with little r, little l, little l, we end up with 100% of the offspring looking like the parents in terms of phenotype. However, if we were to do this cross, in reality we would see something different. We would see something called recombination. So recombination is basically recombining these traits. So instead of just getting red flowers and large leaves and white flowers and small leaves, we would actually have some offspring that had red flowers but small leaves, or some white flowers and large leaves. And why is that? That's because of crossing over. So let's look at these chromosomes here, and let's take them through meiosis. Now, at the end of S phase, the chromosomes look like this. Big R, big L here, and then big R, big L. So this one's copied into the X shape. And then the same with this one here, it gets copied into the X shape. Little r, little l, little r, little l. Now, with no crossing over, what would happen is that this would get separated, these pairs here, and then these would further be separated like that. That's what happens in meiosis. Meiosis 1 separates the homologous chromosomes, and meiosis 2 then separates the sister chromatids. So we'd have gametes that would be big R, big L, big R, big L, little r, little l, little r, little l. However, what actually happens in prophase 1 is we get crossing over, and that's where the chromosomes do this. So there's the chromosome with the dominant alleles on, and it's actually going to overlap with the chromosome with the recessive alleles on. And what happens is that, for example, this might get swapped over. Okay, so the big R here is going to swap with the little r here, just the alleles are swapped. And so the chromosomes at the end up looking like this. Big R, big L. This won't cross over here, and then we get little r there. And then the other one is going to look like big R, so these are basically swapped over. Little l, little r, little l. And so now the possible gametes are big R, big L, little r, little l. That's what we expected here. But now we've got some crossed over gametes. This sister chromatid here is going to have little r, big L, and this one is going to have big R, little l. All right, so let's now do that cross. Okay, the possible gametes are now different. So because of the crossing over, let's do this cross again. Big R, big L, big R, little l, with this organism here, homozygous recessive. Remember, before this cross here, we expected 100% to look like the parent. With crossing over, we get most gametes are going to be big R, L, little l from this organism here. But we're also going to get some switched up because of crossing over. We're going to have some big R with little l, and we're going to have some little r with big L. And these are the crossed over gametes. Now remember, just a reminder, we're talking about linked genes. It's because they're on the same chromosome, okay? So this is talking about linked genes. The only gametes from this organism is big R, little l, because remember, this is the chromosome looks like this. So these are the only possible gametes. So now we're going to have big R, big L, little l. Okay, we're going to have little r, big L, little l. So little l, little l. So that's what we expected before. But now, when we get this here, we're using the crossed over gametes, big R, little l, little l, and now we've got little r, big L, little l. So these organisms here, these two, is what we expected before, like the parents, red flowers and large leaves, and here, white flowers and small leaves. Okay, that's what we expected. However, we now have a recombination of the traits, and that's why it's called recombination. We now have some organisms that have red flowers but small leaves, and some with white flowers but large leaves. Okay, so in an exam, if I ask you, okay, do this cross, what do you expect? Okay, you would expect, because these are going to be the chromosomes here, the cross would be basically big L, little l is one possible gamete, and the other one is big R, little r, and then this one together, little l. They would all look like the parents. In an exam, I say, okay, well, what happens if you now actually do this cross, but you actually end up with some offspring that are either got small leaves and red flowers, or white flowers and large leaves? What's happened? It's crossing over. Crossing over has happened. And if you go back a couple of minutes in this video and watch it again, you'll hopefully understand what's going on. It's because the crossing over is recombining traits. Okay? Alright, so that is crossing over with linked genes. Alright, so the very last thing I want to talk about is the different symbols that we use when representing alleles. Alright, so in normal Mendelian genetics, that's where we just get the mono or the dihybrid cross. We haven't got anything going on with linkage or sex linkage or anything like that. We're going to use letters that represent the alleles. So let's use our example of flower colour. So if you've got big R, little r, and you've got something called complete dominance. Big R is red, little r is white. And if you have big R, or big R, little r, you're going to have a red flower. If you have little r, you're going to have a white flower. Right, now that's complete dominance. In incomplete dominance, think of incomplete like just halfway. So I like to think of it like a cup of coffees. So I like strong coffee, and to make a normal strong coffee, you need 2 teaspoons of coffee. So it's like you've got both the alleles, both chromosomes, are full scoops of coffee. These are meant to be teaspoons. So this might be, let's say, the big C is dominant and little c is recessive. So this will be big C, you've got a completely strong cup of coffee. Right, if you have the genotype big C, little c, it's like you've got just 1 teaspoon of coffee going into your coffee maker and the other one's empty. And so you're going to have half of the strength of coffee. You're going to have a weak coffee. If you have little c, well then you don't have any coffee at all, and it's a terrible morning. So this is incomplete dominance. It's where you've still got a dominant allele, but the dominant allele, you need both versions to be dominant, both alleles to be dominant to get the full-blown genotype. If not, then you're going to get something incomplete. So an example in our flowers, big R would be red, big R, little r would be pink because it's like a diluted red, and little r is white. Now remember that little r is not saying make white, it's saying don't make red, and if you don't make any pigment, well then you end up with a white flower. In codominance, this is where both of the genes are going to work. They're both equally dominant as one another, and a really good example is the blood typing. Alright, so remember the blood types, you've got IA means you're going to make A antigens on the red blood cells. IB means you're going to make B. Little I is none. Okay, so remember you've got 2 versions of every gene. You've got 2 alleles that say make, 2 versions of the gene that say make the antigens on the surface of the blood cells. And there's different alleles you can have for that. If you've got IA, you're going to make A. If you've got a little I, that's going to be type A blood. Okay, so you've basically got either this one or this one for your genotype. You're going to make A antigens. If you've got IA and then the other one doesn't work, or B here and the other one's a B, you're going to have type B blood. If both your versions of the allele are this, they basically say I can't make antigens, so you're going to be type O, which is no antigens. However, if you're IA, IB, well then you're going to have type AB because you're going to make A antigens and you're going to make B antigens. What doesn't happen is that the A is dominant over the B or the B is dominant over the A. They are both dominant. They're co-dominant with one another. All right, so that is that example for normal Mendelian dominance, and then we've got incomplete and co-dominance. Right, there are some other things we've encountered. So you can have genes that are on the sex chromosomes, and the sex chromosomes are either X or Y. Most of the genes you're going to encounter are going to be on the Y chromosome, and males, that's why males suffer more from sex-linked traits, disorders, genetic disorders, because genetic disorders are often a broken allele, caused by a broken allele, broken gene, and normally these genes are going to be on the X chromosome. And so if you've got a recessive allele on the X chromosome, well remember a male is XY, and so he only gets one version of the X. So if he inherits the recessive allele, well then he doesn't have another chance to get a dominant allele to give him the working version. All right. But anyway, the way we write the alleles is going to be the same, but they're going to be written as a little superscript here to the chromosome. So let's look at our example again, where big R is red flowers and little r is white flowers. Well, that means the allele for red flowers, and that's the allele for white flowers. It just means that the gene is on the X chromosome. So if a female is like this or like this, she's going to have red flowers, and if she's like this, she's going to have white flowers. And the male only gets one shot. He only gets one X chromosome. So if he's X big R like that, he's going to have red flowers, and X little r is going to have white flowers. Okay, so that is where we're talking about sex-linked traits, so sex linkage. It's not the same as linked genes. Linked genes are on the same chromosome. These are sex-linked. They're linked to the sex because they're on the sex chromosomes. Right, the last thing, let me make another layer here. The last thing is the Drosophila. So Drosophila have kind of, they have difficult ways of writing their alleles just because of the way that the lab that worked on these alleles decided to name them. All right, so what happens with Drosophila, there's your little fruit flies. We name their alleles after the first mutation that was found. So let's say we're looking at colour, and the normal colour, which is what we called wild type, is grey, and the first mutant to be discovered was black. Well then, normally what we would expect, we have big G and a little g and things like that, but we don't do that with Drosophila. Instead, what we do is we use a plus to indicate the wild type. And the symbol that we're going to use, the letter, is going to be the mutant. And so the letter we're going to use is going to be B for black. All right? So B plus that's the wild type. So that's grey. Just a B by itself is the mutant. And remember, these organisms are going to have 2 copies of every gene, 2 alleles. So if the organism is B plus, or B plus B, assuming that the wild type is dominant, they're going to be grey. And if they're BB, they're going to be black. Okay, so that's how we write the alleles for Drosophila. It's going to be the letter that represents the mutation. So let's pick another trait that we could follow in Drosophila. If it was going to be wing shape, okay, and wild type is normal wings, and the mutant is curled wings, well then we're probably going to use a C. So it'll be C plus is normal, the allele that calls normal wings, and C is curled wings. Okay. Now, the very last thing, and this is the most complex it gets in terms of writing alleles, is that, well, some of these genes in Drosophila can be on the sex chromosomes. And so we're going to do it exactly the same as we did before, where we use a little superscript, but we're going to use this nomenclature because we're looking at Drosophila fruit flies. And so let's say this gene was on the X chromosome. Well, it's going to be like this, XC plus or XC.